Okay, let's jump right into this explainer and explore the fascinating, invisible computer code that dictates literally every structural detail of the plant world. We so often think of plants as these static, simple organisms just sort of passively sitting in the dirt. But let's be real. Beneath the surface, right down at the precise molecular level, they are executing incredibly complex genetic blueprints that rival the most sophisticated software humanity has ever created. Today, we're going deep. We're going to unpack the specific transcription factors, the microscopic cellular engines, and the exact genetic switches that build the botanical world from the ground up.
I want to start off with a pretty provocative question. When you look at the perfect mathematical fractals of a Romanesco broccoli, are you looking at biology or are you looking at a biological algorithm? That mesmerizing beauty, where each tiny bud is just a miniature version of the entire vegetable. Yeah, that's definitely no coincidence. It's the physical manifestation of a genetic program executing a precise loop. See, the plant's stem cells maintain a specific meristematic identity. Through tightly controlled molecular feedback loops, the growing tip continuously attempts to form a flower but fails, and instead it forms another growing tip. And it just repeats that infinitely to create that wild fractal geometry. Think of it like this. Plant shoots are basically running a continuous while loop. Stem, leaf, bud, repeat. Stem, leaf, bud, repeat. These repetitive developmental patterns are completely, entirely genetically determined. And what's truly amazing is that this loop can be easily tweaked by really simple genetic mutations to alter the plant's entire architecture. For instance, a tiny change in the code that controls the synthesis of phytohormones, like auxin or gibberellins, can severely restrict cell elongation in the stem. That exacts a change in the physical space between leaves, totally transforming a tall, leggy vine into a compact, bushy plant. It's basically a simple line edit in the code. Now, to actually zoom in and read this code, we rely heavily on Arabidopsis thaliana. It's a tiny, pretty unassuming weed in the mustard family, but it is an absolute superstar in molecular biology. So why do geneticists absolutely love this little guy? Well, its incredibly small genome, just about 27,000 protein-encoding genes across 5 pairs of chromosomes, has almost no junk DNA. This makes it completely perfect for tracking exact biochemical pathways right down to the base pair level. Because it grows so fast and pumps out thousands of seeds in mere weeks, scientists use it to map the exact blueprints of plant development. Decoding this tiny weed has essentially unlocked the genetic secrets of almost every agricultural crop we eat today.
Section 1. Cellular Mechanics, the Physical Drivers of Growth.
So, before a plant can build complex organs, it has to physically expand its cells. And there is a stark contrast in how different kingdoms of life actually pull this off. When animal cells expand, it is a metabolically expensive process. We have to continuously synthesize massive amounts of new, protein-rich cytoplasm just to fill the space. Plants? Plants are the ultimate economic hackers. They use a massive central vacuole surrounded by a specialized membrane called the tonoplast. By using transport proteins to pump ions into this vacuole, water just rushes in through aquaporins via osmosis. This creates immense turgor pressure, literally filling the cell with cheap water, which accounts for about 90% of their physical expansion. It's this incredible efficiency that allows for staggering growth rates. I mean, we're talking about a bamboo shoot elongating over 2 meters in a single week. But here's the catch. How do plants control the specific direction of this water balloon-like expansion? They use microscopic belts called cellulose microfibrils. Imagine putting a tight, rigid corset around a water balloon. When you pump it full of water, it can only stretch in the direction not restricted by the corset. Inside the plant cell, intricate pre-prophase bands of microtubules act as the architects. They sit right beneath the plasma membrane, serving as physical tracks. Traveling along these tracks are massive cellulose synthesizing enzymes, specifically rosette complexes known as CISA proteins. These protein complexes literally extrude cellulose microfibrils directly into the cell wall, wrapping the cell and forcing it to elongate exactly where the plant's underlying code demands.
Section 2. Genetic Blueprints. The if-then logic of cell differentiation.
Now that we understand the physical mechanism of expansion, let's zoom in even further to the genetic blueprints, the molecular if-then statements dictating what these expanding cells actually become. Like, how does a plant cell actually know its fate? Well, a perfect example of this is the GLABRA2 homeotic gene. This gene encodes a highly specialized homeodomain transcription factor, which functions exactly like a binary switch in a computer program to tell a developing root cell whether or not to grow a root hair. The protein produced by GLABRA2 physically binds to the promoter regions of downstream target genes, suppressing the biological machinery needed to build a hair. The plant needs these root hairs to absorb water for sure, but it needs them in a highly specific, perfectly spaced out pattern.
And the positional logic the plant uses here is brilliant. It's entirely based on cell-to-cell communication. How does an epidermal cell detect what it's touching? It relies on incredible molecular signaling, utilizing receptor-like kinases on the membrane and microscopic channels called plasmodesmata to actually shuttle signaling proteins, specifically WRM and CPC, between neighboring cells. If a cell detects it is touching two underlying cortical cells, boom, the GLAB2 gene turns off and the cell develops a root hair. But if the signaling detects it's touching only one cortical cell, the gene turns on and blocks the hair from forming entirely. The cell's physical location dynamically dictates its genetic fate.
To really grasp the massive scale of these microscopic if-then events, just consider this number for a second. A single 4-month-old rye plant executes this binary GLABRA2 code to grow an estimated 14 billion root hairs. Think about the sheer metabolic computing power required to perfectly execute that binary code 14 billion times. If you laid those tiny root hairs end to end, they would stretch 10,000 kilometers. That is literally 1/4 the length of the Earth's equator, all generated by a flawlessly executed molecular algorithm.
Of course, sometimes there's a glitch in the code, right? And if the initial programming fails, the results are catastrophic. The very first division of a plant zygote absolutely must be asymmetrical to establish polarity, basically the top and bottom axis of the plant. In the non-mutant of Arabidopsis, this code fails. The normal genome protein is responsible for vesicle trafficking, specifically pinpointing auxin efflux carriers to the bottom of the cell to create an auxin hormone gradient. Without that genome protein, the gradient just isn't established. The first division becomes symmetrical, and without that basic up/down axis polarity, the plant develops into this bizarre, rootless, leafless ball of completely unorganized cells. Similarly, tweaking other codes can literally hack the plant's architecture. Take the knotted 1 gene in tomatoes. This is a Hawks-like transcription factor, meaning its main job is to keep cells in an undifferentiated stem cell-like meristematic state. Normally, this gene is turned off as the leaf begins to form. But when scientists actually overexpress this gene, keeping its protein active in the developing leaf tissue, the plant attempts to form new shoots right on the leaf itself. This transforms normally compound leaves into wild supercompound leaves, where leaflets form their own leaflets. It just goes to show how incredibly delicate the balance of these transcription factors really is.
Section 3. The molecular mechanics of flowers, from vegetative to reproductive.
So let's explore the most complex molecular feat of all, the phase change from standard vegetative growth to a flowering, reproductive machine. This is a massive shift in the plant's operating system. How does it know when to do this? It measures environmental cues, like day length, using phytochrome photoreceptors in its leaves. These detect specific wavelengths of light, which then stabilize a signaling protein called constans. Constans sends a chemical signal to the shoot apex, activating Madsbox genes. These genes encode transcription factors that physically interact, binding together to form tetramers, 4 protein complexes that lock right onto the DNA. This shuts down the continuous, indeterminate growth of the stem and starts building a flower from the outside in whorl by whorl.
This brings us to what biologists call the ABC hypothesis. It is an incredibly elegant 4-step molecular sequence driven by those exact Madsbox genes we just discussed. Basically, you have 3 primary classes of these genes, A, B, and C. Their protein products combine in the cellular nucleus to switch on specific organ-building machinery in overlapping combinations across 4 concentric whorls. A alone gives you the outer sepals. A plus B gives you the petals. B plus C creates the male stamens. And finally, C alone forms the female carpels right in the very center.
Now, here's where the logic gets really fascinating. A and C genes naturally suppress each other at the molecular level. They are biological rivals. They actually form repressor complexes that actively block each other's transcription at the chromatin level. So if you mutate the code and suppress the A gene, the C gene totally takes over its territory, resulting in a flower made only of carpels and stamens. Suppress the C gene, the A gene floods into the center, creating a mutant flower of only sepals and petals. By knocking out just one single variable in this molecular code, you get beautifully bizarre, completely misshapen mutant flowers. To wrap up this explainer, I want to leave you with a massive question to think about. We've taken an incredible molecular journey today, from the turgor pressure of a single expanding vacuole all the way to the DNA-binding Madsbox tetramers that build a flower. We are no longer just observing plants, we're actively reading their source code. As we master these in-depth molecular details, manipulating everything from cellular transport proteins to complex transcription factors, could we engineer entirely new, climate-resistant crops to save our planet? The invisible code is right there waiting for us, and honestly, our agricultural future might just depend on it.